little bit on getting primary literature. That has nothing to do with that paper you're holding, by the way. Everyone gets 25 stars, good job. So, 2 ways that I find literature. When I'm searching for literature, when I have a topic I'm trying to learn about, these are the 2 places I go. I personally go to PubMed first, even though I listed it second. They're both search engines for doing searches. The Madden library I use more to actually download references of PDFs of references that I can't get for free at PubMed. That's the way I do it, not the only way to do it. Okay, so quickly, if you go to the Madden library website, I'm on a computer, I suppose I could actually go to the real website, but I took some screenshots instead. So it'll look like this. You click on the journals and newspapers link, and then there's a search field, and you can type in whatever it is you want. Normally, information from a citation. I usually use last names of authors. If I know what, so remember, I go to Madden Library when I have a paper I'm trying to get the PDF of. And so I already know the citation. I know the author's names, I know the journal, I know the year, I know the volume number and all of that. So maybe I type in author names. In this case, I'm typing in the name of the journal, because I want to know, does the Madden Library have online access to that journal? So we just read a paper from Genome Research. So I start typing in Genome Research, and I get to RES, and it tries to autofill. There's one journal that matches that criteria, so I click on Genome Research. There, hit search. And it comes up with this record, one electronic journal, and full text online is what I want to see. Free, I really like to see. Although if you're logged in, so you have to be logged into campus, and this may or may not work for you, by the way. I've heard some students say that they don't have as much access as faculty do through this resource, so try it, see how well it works for you. 2 different links to get the full text of articles from this journal, Genome Research. And it tells you there that this is always the case and annoys the hell out of me, but we can talk about that in later weeks, why that is, not why it annoys me, but why it is that most recent 6 months are not available. So if you just saw that brand new paper and it has everything to do with your research and you want to get it, if you want digital, you have to pony up the cash, pay for it. When that window pops up and says, pay us $108 for 24-hour access to the, whatever, I don't know, I've never paid the money. Yeah, just to access, yeah, so if you buy,
24 hours?
Don't get me started. Seriously, folks. But available from 1991, volume 1, issue 1, through 6 months ago. This journal. Some journals, the day it comes out, it's available free online. Some journals, there's a 6-month embargo. Sometimes, who knows? Okay, so you could click on either of those links, presumably, and it would take you to a journal website where you could download the information. That's how I use the Madden library. It takes you to the journal website, and you have to navigate through the journal's website to find the volume, issue, article. Then there's PubMed, which I like more because I use it to do free text searches. I've got not a journal that I want to search for. Does the library have this journal? I want to know, I don't know, did I put an example in here? Oh, so I searched for our author, Dr. Anita Zinsky, senior author on this paper we just read. Do these look familiar? Yeah, number 6. So if you click on the link, this is the PubMed results section. So every time it has a hit that matches your search terms, it shows it to you. You click on the link, the title, that takes you to the record page, the entry. So if I click, so you get the title again, list of authors, the abstract, which is sometimes all I want. Sometimes I just want to see the abstract. So this abstract's always free, regardless of whether or not you have to later pay to get the whole paper. More information about the citation. So again, if you were searching on the search terms replication origin activity, this would come up. Then you'd have the full reference. Stick that in your literature cited section after you read the paper. Genome Research, 2012. October, volume 22, issue 10, pages 1953 to 62. Go ahead.
Oh, just a quick point, you say, put that in your reference section after you read the paper.
Yes.
Very important, after you read the paper.
That's right.
Because you don't necessarily know if you're going to agree with the author's interpretation of the data that they present in the abstract. It can be really tempting to read the abstract and go, cool, that relevant.
Sounds right.
That sounds right.
Yeah.
I'll stick it in my reference list and move forward.
Yeah.
It is tempting. And anybody who tells you they haven't been tempted like that is lying. But do read the paper. It is important to gain all the subtleties of how they did the work.
Yes, absolutely. More than that, don't just base it on the abstract, especially don't do it just on the title. Guilty. What I'm really looking for and why I go to PubMed is those links. That's where you go get the PDF, the actual article. What I'd love to see is free. Full text Genome Research, free. That's the journal, so the website of the journal Genome Research. You click on that button, it takes you to the journal, you download the PDF. The nice thing is that the US government a while back got a lot of frustrated customers, citizens, complaining about the fact that they were paying tax money that was being used to fund National Institutes of Health Research, National Science Foundation research, and other government agency research, USDA, etc. But had to pay $108 for 24-hour access to see the taxpayer-funded data. So then they say, okay, there's a problem. So if now, and for the last decade or almost decade now, if you're doing research in the US and it's funded at all by government grants, you have to make the data freely available, and any publications that come of it. So that's where this free and PMC is PubMed Central. So if you're an author on a paper that's funded by federal tax dollars, you deposit on your own a free copy in PubMed. That's why I go to PubMed. Because if you just see a citation and it says go to Nature, the journal Nature, nothing is free at Nature's website. But you can go here and access that free link. Not for every paper, but for a lot of papers, especially if they've been done in the last decade or earlier.
I'm assuming because it's on PubMed, just because it's private domain, and therefore they would charge you?
Wait, what?
Well, you're saying like on Nature.
Yes. Well,
I mean, obviously not for PubMed, because...
So Nature, yeah, is for profit,
Yeah.
and they charge for everything. Libraries pay to have access to Nature journals. So that's one reason to go through. So if you go to PubMed and it says it's not free, well, if you go to PubMed and it's in Nature and it's not free because somebody in another country did it, they don't have to make it freely available. The US government's not telling them anything.
They can't.
Then you go to the Madden library and see if you can log in, and because the campus might might pay for access to Nature. So you log in through campus, through the library, then maybe you get it for free. I always check PubMed first because there's a lot of free literature out there. More of it is available through PubMed. And this is just a database. This is not aside from PubMed Central, this is just a resource for searching. Lots and lots and lots of journals.
Even if you do plant stuff like I do, PubMed
Oh, really?
is the first place I go. I don't find as many, but it's a good lead-in.
Yeah, there are other databases for looking for, and they're particular. I'm an evolutionary biologist. PubMed does not index the journal Evolution, the journal in evolutionary biology. This database has lots and lots and lots and lots and lots of journals that I read. There's just that one stands out, and it's because it's not necessarily biomedical. PubMed is part of the NIH, National Institutes of Health. So they have a little bias there. Okay. I think that's about it. Oh, one other thing that I find really important that I love to pass on. When you see a citation that has some horrible abbreviation of a journal, you have no idea what the hell that means. What is Proc Roy Soc Lund? You see that in the short little bibliography. Author names, year, journal issue number and then this. How do you find out what that means, and how do you find the journal that that belongs to? If you go to NCBI, to PubMed, use this drop-down menu. One of the options is, there's like 50 things in this menu. It's huge. One of them is PubMed. Go to NLM Catalog. Type in whatever crazy abbreviation of a journal you've got. Procroy Soc Lund. And it gives you matching results. So I clicked on Proceedings of the Royal Society of Medicine. It tells you everything that there is, or practically everything there is to know about that journal, including all of its various abbreviations. And so that can be really useful when you run into a journal that you have no idea what the abbreviation stands for. Search the NLM catalog. Okay. Oh, a couple other points really quickly, and then we're on to a little bit of information for next week. When you go to that abstract page, a really, really useful way to get more information that's written for a lay audience, good thing to read first, if it says comment in, that's telling you there's a link that's related to this publication. That is, for example, in Science, somebody made a comment, like a one-column brief synopsis of the paper, usually really good things to read first, like a summary, like an abstract of the abstract. They can be really useful resources. Another thing to look on this page in particular, whether or not this paper has ever been retracted. Don't read it if it's been retracted. No one's going to believe you if you cite it. Okay. So we'll talk about retractions another week too as part of this time period. What it takes to get retracted, what it means, implications, how it works, and so forth. The fact that you never want your paper to be retracted. Okay, and lastly, reviews. So we talked last week about inversions in fish. So if you wanted to, chromosomal inversions. If you wanted to find a review paper, something that synthesizes lots and lots and lots and lots of literature on chromosomal inversions, just add review, the word review, to your search. And then look for that bold thing that says review. Good ways to get lots of information, again, written for more general audiences. They're really useful for finding citations for reading, not citations for just putting in your bibliography, but citations for reading for the GWR, or for a manuscript or anything else. Unless you speak French, avoid the French journals there. Okay. How else do I find journal articles? Wikipedia. So when I don't remember which order of the different phases of the cell cycle I go to Wikipedia. Don't cite Wikipedia, go to the bottom of the page and find the primary citations. Right? Another way to get literature, especially basic literature, because usually the first part of the introduction of a paper, GWR, manuscript, whatever, is really basic stuff. And you sometimes scratch your head and think, wait a second, what am I going to cite about DNA as deoxyribonucleic acid that's made up of 4 nucleotides? So then maybe I'd go to Wikipedia, because almost certainly some really detail-oriented person has put all these references in to the most simple things. It's a really easy way to find resources, references. Lastly, for papers that I cannot find free through the Madden Library, on PubMed, Google Scholar even, I Googled the corresponding author on the paper, usually the senior author, the faculty member. Just Google their name. Find their website. Because on Konrad Niedezinski's website, there's a tab for publications. And if you click on that, he probably has a link to the publication. Not always free, it depends, it's case by case, but this is usually a good last resort. If you go to my website, I have a publications link, and I have all the PDFs freely downloadable. I'm not PubMed, I'm not the Madden Library. It's probably, in some cases it's illegal, and if I ever get caught, I'll probably get in trouble, they'll just make me take them down. But
another thing you can also do, and why it's handy to build up a reference, your contact list, is I still have friends, at University of Illinois. I'll contact them if I can't get a paper. I'll say, hey, Marmos,
Yeah.
I need a copy of XYZ, we don't get it.
Yeah.
Can you, send it my way? And once they have the PDF, then it's easy, they just send it to you.
Yeah,
So there are ways around in terms of utilizing various resources and connections.
User network, yeah.
That was my question. I was gonna ask if it was taboo to ask for a copy.
Well, and of course, the official version of doing that, although maybe another week, should make a note, I'll speak into the microphone. Maybe another week we can talk about information copyright. So there are ways for academics to argue that they should be able to use copyrighted material and distribute it to their classes freely. And so whether or not it holds up in court, sometimes it does, sometimes you still get in trouble. The legal version of what Dr. Constable's, well, more legal, I guess, less problematic, would be interlibrary loan. So you can ask the library to find out if other libraries in the CSU system or the CSU plus the UC system, maybe, I'm not sure how broad the interlibrary loan network is. Previous institutions I've been at, you say, here, library, send them an email, or there's a form to fill out probably. I've never done it here. Here's the citation, please find it for me. Other places I've worked, 24 hours later I get a PDF in my inbox. They've sent it to an institution that we've got an agreement with. They had it, they scanned it. Somebody working at the library went and pulled the journal off the shelf, scanned it, put it in a PDF, and emailed it to me. So sometimes, but you never really know how long that's going to take. So if you're in a rush, well, never be in a rush. Always do things well in advance, of course, and life will be fine. Always do things well in advance of knowing that you need to do them. All right. Okay. All right. Let's do this paper thing first. You've probably already read it. So let's do it. So each star is a protein complex. The lines are chromosomes. We've got the same chromosome from 6 different individuals. So the question we're going to ask is, are the distributions of these stars random across the chromosome or not? And if they're not, do you think it's the case that 2 stars are not really close to each other because 2 stars are not allowed to be near each other on the chromosome, 2 protein complexes? So how, if you got these data, again, thinking like the scientist, some pictures of chromosomes and here are the positions of protein complexes. The sizes don't matter. Just some are different sizes than others. How would you do that analysis? How could you quantitate these data?
The spacing between the protein complexes, perhaps?
Okay, yeah. So you measure, what would you measure? I mean, be more specific is what I'm saying. So you measure the space between the stars.
Yes.
Okay. Space between the edges of the stars, space between the centers of the stars. If 2 stars are touching, basically what I'm asking is if 2 stars are touching, is that 0 distance or is that still some amount of distance?
Well, that hasn't been, technically that hasn't been explained.
Right.
Described, so
No.
until you know that, you can't actually quantify it. If you're going by the tips, then you measure from tip to tip, but if you're going by center,
Okay.
But wouldn't tip to tip and center to center be the same thing?
not necessarily.
Depends on.
If you're comparing the same size stars, they would be. If they're different size stars, they both wouldn't be.
As long as it's consistent.
Perhaps.
Might not work.
You can define your own methodology.
Yeah, that's what
As long
I'm
as you're
saying.
consistent.
Yeah, exactly.
Yeah.
Sorry, Joe.
No. That's the point. So which of these would be the easiest? So let's say we're going to measure something about the distance between the stars. What are some of the differences in the chromosomes? Yeah, just like that example we just saw. It's like Cerevisiae and Bayonus and some other ones. The lengths of chromosomes look different. So if you measure the distance between the stars, does that give you good information about their positions?
I would go more with almost like a percentage type way of looking at it.
So you measure the,
So each one's 100%. If one's directly in the middle, it's at 50% of the length of the chromosome.
okay.
That way you
So
can
that's just
compare them.
relatively speaking.
Yeah, so you measure the distance and then you divide it by the total length of the chromosome to get the fraction of the chromosome length that's in between two stars. Something like that. Which chromosome would be the easiest to measure if you number them 1 through 6, top to bottom? Which one do you think would be the easiest one to do this sort of analysis?
Number 5.
Okay. Why number 5?
They look pretty even, but I guess it's a visual thing, not actually.
So they're
It's more
evenly
quality than quantity.
spaced,
No overlap.
no overlap. Right, none of the stars are touching.
Size of the stars are similar.
Similar star size. Would it be better to have, if they weren't overlapping, would it be better to have bigger stars or smaller stars? Which would give you a more accurate measurement?
Well, how does star size affect the actual chromosome distance?
Depends how you're measuring.
So let's say we're going to measure from the center of the stars.
Okay.
Does it matter how big the star is? You can still figure out where the center is, regardless of how big the star is.
Right.
Okay. So the only thing that really matters about the size is the bigger the star gets, the more likely it is to start overlapping adjacent stars.
But the center should be the same.
The centers will be the same, so we should be good.
So if you go by center, that shouldn't be as near.
Okay,
If I can make a comment,
please.
shouldn't you, if you go to chromosome 3, far right, you've got the biggest star is just to the right of one of the smallest stars. So if you can't just ruler center to center, you've got to correct for differences in height of the center off of the chromosome.
Ooh. I guess,
So you have to measure parallel to the chromosome from the central point of one star to the central point of the other star.
absolutely. Yeah. I should have centered them all on the
Measure a bunch of
chromosomes.
stars.
Okay, so I've done some of the work for you. I've normalized all of the chromosomes to be the same length. Projected onto Cerevisiae coordinates.
Fancy.
No, this is not Cerevisiae, but it doesn't matter what it is, I guess. Okay,
This is Rossi
this is, yeah, that's right. If I may ever be so lucky as to have a species named after me. Okay, so here's what I propose we do. What I did was I took this figure and I wanted to make even divisions, so I folded it. So I folded one edge along the left edge of the chromosomes. I folded the right edge, so I'd like you guys to do this with me. Fold the right edge along the right edge. So you're just looking at the entire width and nothing more of the chromosomes.
Okay.
You're asking OCD people to make very careful folds.
Yes, I am.
Great.
I used my metal ruler, and the only reason for doing this really is to make even divisions. Right, so we can estimate fractions of the chromosome.
This is enough for people like
Okay. Okay. So one way, again, thinking about how you collect potentially visual data and then translate it into something that's quantitative. Measure what fraction of the chromosome from the left end down you see a protein complex. So next week, we're talking about synapsis and meiosis. Doing recombination as our topic next week. And so we have to talk very briefly now about synapsis and meiosis. Sorry, you're not looking at what I'm looking at. next week. So we got to talk about synapses and meiosis briefly, just an introduction, remind you what that's about. The concept of crossovers, and two types of interference. And again, this is being recorded, so I'll just upload this to YouTube if you don't want to take notes at the moment. Two types of interference. Has anybody heard about interference before? You remember that? Probably talked about it many years ago in a genetics class at some point, maybe. So remember what meiosis is. Production of gametes, haploid gametes on this end, starting at a single diploid cell. Where does synapsis happen? What is synapsis? Do you know where else do you hear about synapses?
In the neural system.
What is a synapse? That's where two cells
Well, two neurons create a junction.
come very close to each other.
Yeah, dendrites and
Yeah.
axons.
So that's so synapse and synapsis are related in the fact that it's things coming together. So this is synapsis. So there's replication. Each of your two copies, the copy of the chromosome you got from your mom and the copy you got from your dad, replicate. Those are sister chromatids. And then the pairs of sister chromatids synapse. That's this stage, metaphase 1. Okay, so that's synapsis. We're talking next week's paper about synapsis. This is what synapsis looks like in a cartoon form. Those two sister chromatids, the copy of chromosome you got from your mom and the version that just replicated, the copy, are the maternal in this case. So you get two different, each of these loops is meant to represent one double helix of DNA. So those two sister chromatids are very tightly intertwined, and they're attached to a protein complex, the axial core. And then the two copies, the sister chromatids from your dad, do the same thing. They're closely intertwined with each other, and they're also connected to an axial core. The synapsis comes from the zippering up using these proteins of those two axial cores. So you make a protein bridge between one dyad, two sister chromatids, and the other one. That's what holds all four of those things together in a tetrad of meiosis 1, metaphase 1. Okay. The point of this is to note that there are proteins involved in synapsis, the bringing together of the pairs of sister chromatids. This is what it looks like in real life. So these dark things are the two axial cores, and emanating are tons and tons and tons and tons of loops of those intertwined sister chromatids. This is early meiosis, before the chromosomes are really condensed. They're mostly condensed here, but what you're looking at in black there are those proteins, proteinaceous structures that run the entire length of the synapsed chromosomes. So there are 4 different sets shown here. So proteins are essential for synapsis. Okay. The proteins that are involved in initiating synapsis. It's a horrible title, I don't know why I wrote it like that. There's synapsis initiation complexes. Everything is something initiation complex. Replication initiation complex, etc. These are synapsis initiation complexes. A set of proteins that exist where those sister chromatids and pairs of sister chromatids are first brought in close opposition to each other. And that's where that zippering up process starts. So a core complex of proteins binds the pairs of sister chromatids together, and then that protein that cross-links the axial cores comes in and just goes along the chromosomes and brings the rest of them into synapsis. The protein in yeast that does the zippering, this transverse filament, is called ZIP1. That's one of the proteins that will be in the paper next week. The synapsis initiation complex has a bunch of different proteins in it. In yeast, it includes ZIP2 and ZIP3. So one of the problems with yeast is all the proteins and genes are named after their function, and so you get ZIP1, ZIP2, ZIP3, ZIP4, ZIP20 probably. They're all different proteins, they do different things, they just have very similar names. So the ZIP2 and ZIP3 proteins are found in these 6 synaptonemal initiation complexes. And ZIP1 goes the whole length of the synapsed chromosomes. Okay, so here is fluorescence of ZIP1. So you can't see the chromosomes right now, but this fluorescence is in between two pairs of dyads. You get dyad fluorescence in between the synapsis, the synaptonemal complex, all these proteins, zip one, and then the other pair on the other side. You just can't see the DNA because it's not stained. So this is that, all those proteins that go across from the two axial cores, all the way down the chromosome. So there's chromosome all along one side of that, what is that, red? Whatever color this is. Down one side of that is one pair of sister chromatids, and along the other side is the other pair.
Is there one sick per synapse?
Ah,
Sorry.
fabulous. Good timing. So per chromosome, there are lots of synapsis initiation complexes. So in this figure, for example, you see two. There could be lots. So synapsis initiates along all of the chromosomes, much like yeast arses. There are lots of positions where replication starts. There are lots of places along each chromosome where these what are we calling them? Synapsis initiation complexes form. So they can form at multiple places, and then you get the zippering up in between them. So the concept just simply is there can be multiple locations of synaptonemal initiation complex. Okay. Why is this important? So there's synapsis, the bringing together of the chromosomes. That's one cellular event. Then there's genetic crossover, which is why meiosis 1 is really important. Dredge it back up from your memories if it's been a while, but this is where strands of the different sister chromatids, once they've synapsed, break and exchange DNA segments. You recombine. You make new combinations of alleles on chromosomes that didn't exist before. You pass those on to your kids. That's one of the main purposes of meiosis. Produce gametes, and then synapsis, in this case, produces genetic variation. I'm just reminding you. That's not entirely critical for the paper. But this is what that looks like. We replace the cartoon with an actual micrograph. You can see this is 4 sister chromatids, some of which are crossing over, exchanging genetic material between them. So you can see a chiasma, or the crossing, there, and there. So this set of sister chromatids is exchanging 3 different times. Aside from creating genetic variation, what else does this do? The physical process of recombination, crossing over, does something really important too. It holds together the tetrad temporarily during meiosis. Talked about, okay, so interference, last thing. Good. Is it random where those chiasma, the sites of crossing over, occur? Do they happen all along the chromosome? As we discussed briefly recently in Caenorhabditis nematodes, they happen very rarely in the middles of chromosomes, more frequently on the ends. That's more of an exception rather than a rule. In humans, recombination happens all over the place on chromosomes. There are some places that happens more frequently than others, recombinational hotspots. So
So it varies between species to species?
So as it varies from species to species, or genus to genus, or family to family, or something like that. But one way that crossovers are not random is that when you have a crossover happen, the likelihood that there's another crossover very nearby is much lower than the likelihood of having another crossover farther away. So this is more frequent than you would see 2 crossovers very close to each other on the chromosome. This is called interference. This crossover is interfering or pushing other crossovers far away from it on the chromosome. Don't know how. That's the big question for next week's paper. We still don't really well understand how interference at the molecular level works. We've observed that this is much less frequent than this. So we've got evidence that this happens, but how exactly is the information that there's a crossover happening here passed on to the rest of the chromosome? How is it that the chromosome knows how to spread out the crossover events? Now, that's genetic interference, because these are genetic events, the actual recombination of different molecules of DNA. Then there's what the authors of this next week's paper are going to call cytological interference. The synaptonemal initiation complexes also exhibit interference. That is, if you look along a chromosome, this might start looking familiar, if there are protein complexes where we can actually observe the locations of the SICs, maybe by looking for proteins like ZIP2, can we observe interference among these proteins? That is, if we see one here, is it more likely that the next one is far away on the chromosome than close? There's no known yet, there is no known link between cytological interference, whether these are spread far apart, and whether or not these have anything to do with the locations of crossovers. So as far as we know now, this and this are totally different. Where the chromosomes start synapsing doesn't necessarily have anything to do with where the chromosomes cross over. They could be two totally separate things. Because remember, this has to do with bringing the chromosomes together, just where that process starts along the chromosomes. This has to do with chromosomes breaking and exchanging genetic material. They could be two totally separate things, or they might be intimately related. Don't know. So this paper that we're doing next week aims, in essence, to find out. So this is exactly what they're going to do next week. I've described it briefly here on the board. Here's the approach. This top part is ZIP2 fluorescence. So they've used an antibody, I think, to ZIP2 protein, so they can see that's the outline of a chromosome. The bottom here is ZIP1, so that gives us the length of that chromosome. Right? This is the fluorescence of the protein core that goes all along the synapsed chromosomes. So that gives us length. We've got ZIP2 fluorescence. So presumably these blobs in the top picture, how many blobs do we have? Like 1, 2, 3. Can't tell if that looks like it might be two next to each other. So we've got the total length of the chromosome measured by ZIP1 fluorescence. And we've got the relative locations of blobs of synapsis initiation complexes on the top. Do those exhibit interference? Are they evenly distributed? Do we find some cases where, so what they've done here, I've just cartooned it. They divide each chromosome into 10 different equally wide segments, and then they just catalog in what segments are the different SICs, the ZIP2 blobs. And so this is the main type of figure you're going to see next week, where we're looking at those 10 different intervals of equal size, 1, 2, 3, 4, up to 10. And then the percent of total foci, that is the fraction of the total blobs on a chromosome that are found in each of those tenths of the chromosome. So, on average, 9% of ZIP2 containing synapsis initiation complexes are in the first tenth, 14% are in the second tenth of the chromosome, and so on. That's what you're going to be looking at next week, a lot. And we're going to find out, do these proteins exhibit interference? Are they evenly distributed along the chromosome? This makes it look like they kind of are. You're almost equally likely to see one of these protein complexes in any part of the chromosome. Do they exhibit interference, and do they have anything to do with where recombination happens? And with that, I'm happy to release you into the night. Questions? Yes, please, of course.
Does that mean crossing over doesn't necessarily have to occur during the synapses occurs?
Does crossing over have to occur when synapsis occurs? Yes.
Does it occur all the time when synapses?
There has to be at least one crossover per meiosis, per chromosome. there's always one event that bridges those two pairs of diehads. There has to be some sort of recombination. Okay. Other questions? Great. So the paper for next week is already up. And, yeah. Happy reading when it's time to read for next week.
I have a quick question.
Yeah.
Does interference, just because it happens, does that mean, or does it always mean that a recombination can't happen closer, or is that just kind
Well,
of?
that's the question, is there anything is there a relationship between where synapsis starts and recombination?
Okay.
So go into it agnostically, just assume that there isn't, and then read the paper and see what they say and what evidence they have.
Okay.
Yeah.
Can I know when your office hours?
My office hours, Monday and
I
Wednesday,
sent you
something and
mail,
something.
but
and-
I sent you a mail.
Yes,
Regarding like asking about your research.
Yes,
you
and I
asked me, like come meet you, but I